The meeting between Homo sapiens and Homo neanderthalensis is one of the great unresolved stories of our past. We know it happened. The evidence lives in us. Every non-African human carries traces of Neanderthal DNA in their chromosomes. Yet, two crucial inheritances are missing. No Neanderthal Y chromosomes, no Neanderthal mitochondrial genomes. The genetic record whispers of intimacy, but also of erasure, of contact that left fragments, not families. How could Neanderthal genes enter our lineage but leave no direct paternal or maternal line behind? That mystery has a way of steering the imagination. Most reconstructions start from a familiar picture. Modern humans expanding out of Africa, displacing the Neanderthals they met along the way. Within that frame, it feels natural to assume that interbreeding, when it occurred, mostly involved modern men and Neanderthal women, the conquerors and the conquered. It is a vivid image, and that is part of its power. But vividness is not evidence. The biology of hybridization adds another layer that most narratives overlook. In many species where two related lineages meet, hybridization succeeds in only one direction. Differences in fertility, behavior, or immune response can make one pairing viable and the reverse impossible. This pattern, well established in evolutionary genetics, means that one sex combination can produce offspring that thrive, while the opposite pairing fails. In the case of humans and Neanderthals, that rule should make us cautious. If only one cross worked well, our reading of the genetic traces must begin with the question, which one? Here is where the two blind spots meet. The prevailing assumption of modern male and Neanderthal female unions aligns neatly with the common hybridization bias, but for the wrong reasons. It blends a biological inference with a social expectation. The result is a self-reinforcing story. The imagined conquest supplies the direction of mating, and the direction of mating is then used to confirm the conquest. What gets lost is the possibility that the asymmetry pointed the other way, that Neanderthal men, not women, were the primary contributors to the surviving gene flow. Once that door is reopened, the pattern in our genomes looks very different. Ancient DNA has given us an outline of what happened when these two human species met. All living non-Africans carry roughly 1 to 3% of Neanderthal ancestry, yet no Neanderthal Y chromosomes or mitochondrial lineages survive. The question is how those three facts, autosomal persistence, paternal loss, and maternal absence, can coexist. What kind of contact could have produced them? The simplest answer begins with two forces that we can test directly: a directional bias in mating and a biological filter that acted more strongly on males. If most successful pairings joined Neanderthal men with modern human women, then modern mitochondria were transmitted from the start. If hybrid males faced reduced fertility or immune incompatibilities, their Neanderthal Y chromosomes would have vanished within a few generations. What remained, the fragments scattered through our autosomes, survived because they were neutral or beneficial. One demographic asymmetry and one selective effect explain the entire pattern. That, in essence, is our thesis. The genetic legacy of the Neanderthal encounter is best understood as the outcome of predominantly Neanderthal male and modern female admixture, shaped by sex-specific selection against hybrid males. This framework does not glorify one species or condemn another. It simply fits the evidence with fewer assumptions. Today we will trace how social behavior, reproductive biology, and evolutionary constraint together forged the quiet but enduring imprint of Neanderthals within us. To understand how Y and mitochondrial lineages behave, we must first recall that they are not neutral recorders of ancestry. They are shaped by the social structures of the people who carry them. Across global populations, The Y chromosome tends to be far more geographically differentiated than mitochondrial DNA. In other words, Y lineages tend to cluster locally, while mtDNA shows broader geographic continuity. This was first demonstrated in classic studies by Seilstad and colleagues, and the explanation is profoundly human: patrilocality. In most societies, women move more often than men, marrying out, joining new communities, carrying their mtDNA across social boundaries, while men remain within their natal groups, transmitting their Y chromosome locally. This difference alone creates a global pattern. Y diversity becomes patchy and regional, mtDNA more homogenized. Where this pattern reverses, such as in matrilocal societies, the genetic structure reverses too. The Y becomes more widespread and mtDNA more localized. From this, we learn that uniparental genetic geography reflects sex-biased social behavior, not inherent molecular properties. For our purposes, this means that the Y chromosome is an especially sensitive instrument for recording male demographic shocks, wars, migrations, or replacements. Over long timescales, repeated male-biased events can wipe out Y lineages entirely, while the mtDNA, transmitted through women who move and intermarry, may appear comparatively stable. The contrast between the mitochondrial genome and the Y chromosome should be read as a record of social pathways rather than as a property of molecules. When women move and men stay, maternal lineages behave like a solvent that blends neighborhoods, while paternal lineages behave like dyes that stain local groups. Reverse the social pattern, and the genetic picture reverses. In the deep past, this social lens gives us a disciplined way to infer directionality in contacts that left no written record. If the Y chromosome seems volatile, that is because history shows it to be so. Two well-established mechanisms explain why. First, there is what geneticists call the post-Neolithic Y bottleneck, around 5,000 to 7,000 years ago. During this period, global Y diversity collapsed, while mtDNA diversity remained high. The most plausible explanation is patrilineal competition. As societies grew larger and more hierarchical, reproductive success became concentrated among dominant male clans. The Y chromosome of a few successful groups expanded, while those of others disappeared. This was not due to biology, but to social structure, a genetic echo of cultural dominance. Second, ancient DNA has revealed multiple episodes of male-biased migrations and conquests. During the Bronze Age expansions from the Eurasian steppe, for example, invading males almost entirely replaced local Y lineages across much of Europe, even though autosomal and mtDNA contributions from local populations remained substantial. The lesson is clear. Y lineage replacement can happen suddenly and completely through demographic events alone. Thus, when we infer that the Neanderthal Y disappeared after contact with modern humans, we are not invoking anything extraordinary. We are observing a pattern that has repeated many times within our own species. The absence of Neanderthal mtDNA has often been treated as a separate mystery, but it too can be explained by well-understood processes. First, if the majority of successful unions were between Neanderthal males and modern human females, then every hybrid child would have inherited its mitochondrial genome from the modern mother. The Neanderthal mitochondrial genome would simply never have entered the long-term gene pool in any quantity. Directional mating alone is sufficient to erase it. Second, even if a few Neanderthal mtDNA lineages did enter early human populations, they would have faced a strong headwind from mitonuclear incompatibilities. Mitochondrial genes work in tight coordination with nuclear genes to manage cellular energy production. When lineages that have evolved separately for hundreds of thousands of years mix, Mismatches between these systems can reduce fitness. Studies across animal species show that hybrids with incompatible mitochondrial and nuclear combinations often suffer reduced fertility or viability. Natural selection quickly removes the discordant mtDNA. Thus, even rare Neanderthal mitochondria could have been purged through ordinary biological selection. Third, mtDNA lineages are not immutable. Archaeogenetic time series reveal entire turnovers of maternal lineages during the Neolithic transition in Europe. Some haplogroups that were once common vanished completely within a few thousand years. This demonstrates that maternal lineages can be replaced when demographic or cultural currents shift. Together, these points dissolve the mystery. The lack of Neanderthal mtDNA today follows naturally from directional mating and known selective dynamics. There is no need to appeal to sweeping or improbable forces. A small initial frequency of Neanderthal mitochondria caused by directional mating, compounded over many generations by ordinary selection for mito-nuclear harmony and by the random pruning of maternal lines through drift and founder events, is fully sufficient to drive those mitochondria to practical extinction. The same logic explains why rare introductions can flicker and vanish even when autosomal traces endure. The mechanism is mundane, which is precisely why it is powerful. Let us now place the three key observations side by side. Modern non-African populations contain 1 to 3% Neanderthal DNA in their autosomes. No Neanderthal mitochondrial DNA survives. No Neanderthal Y chromosome survives. To explain all three, we do not need three separate events. We need one coherent demographic and biological process. If the encounters between the two species were asymmetrical, with Neanderthal males mating with modern human females, then the hybrid offspring would automatically carry modern human mitochondrial DNA. If, in addition, male hybrids suffered reduced fertility or viability, whether because of immune incompatibilities such as the HY antigen system or more general reproductive mismatches, the Neanderthal Y chromosome would fail to persist. Finally, autosomal segments, those not tied to uniparental inheritance, would continue to diffuse through backcrossing, accounting for the small but measurable Neanderthal fraction in all non-African genomes. This single pattern of asymmetric mating plus male-specific incompatibility explains all three observations in one stroke. It is the simplest, most economical model available. By contrast, if interbreeding had been symmetric, if Neanderthal females and males both contributed equally, we would have to imagine two separate and powerful selective sweeps, one to eliminate Neanderthal mtDNA and another to eliminate Neanderthal Y chromosomes. That would be far less parsimonious. The genetic evidence aligns precisely with this asymmetric scenario. First, there is the consistent depletion of Neanderthal ancestry on the X chromosome compared to the autosomes. This pattern can arise both from male-biased admixture, fewer Neanderthal X chromosomes entering the modern gene pool, and from selection against male hybrid incompatibilities linked to the X, as predicted by Haldane's rule. The most conservative reading of the X chromosome evidence is that contact was both sex-biased and male-costly. Depletion on the X relative to the autosomes follows naturally when Neanderthal genetic input arrived mostly through males, and when male hybrids were less likely to leave sons, who in turn left sons. The observed deserts around testis and meiosis-related genes are not a curiosity but the expected footprint of such a history. In this light, the dual absence of Neanderthal Y and Neanderthal mitochondria is not two mysteries but one coherent outcome of who mated with whom and which lineages could amplify thereafter. Second, the deserts of Neanderthal ancestry in modern genomes are concentrated around genes expressed in the testes and those involved in meiosis. These are precisely the regions we would expect to be affected by reproductive incompatibilities between species. And third, of course, is the double absence. No Neanderthal Y, no Neanderthal mitochondrial genome. Under our model, this is not a coincidence but a natural outcome of the same demographic skew. Let us pause to reflect on how the pieces fit together. The mating pattern determines what enters the hybrid population. The biological compatibility of the sexes determines what survives within it. And the stochastic forces of culture and demography determine which lineages expand or vanish thereafter. When we combine these, we find a self-reinforcing logic. Male-biased mating brings Neanderthal genes into the human population through females who carry modern mitochondria. Male hybrid incompatibilities remove Neanderthal Y lineages. The autosomal fragments that remain survive because they are either neutral or beneficial. Each of these processes reinforces the others. None stands alone. Taken together, they form a complete explanatory circle. There are several avenues by which this interpretation can continue to be tested as ancient DNA archives grow. Comparing the X chromosome to the autosomes can provide direct estimates of sex bias in admixture. The degree of Neanderthal ancestry difference between these regions can be translated into the proportion of male versus female Neanderthal contributors. Examining nuclear genes that interact with mitochondrial functions may reveal whether archaic variants are specifically depleted near these loci, as would be expected if mitonuclear incompatibilities played a role. Temporal series of ancient genomes from contact zones could show the earliest hybrid individuals carrying modern mtDNA but lacking Neanderthal Y chromosomes, providing a direct snapshot of asymmetric mating and selection in progress. Each of these predictions stems naturally from the logic of the model. None requires ad hoc assumptions. When viewed in light of these principles, the pattern of Neanderthal ancestry and modern humans is no longer a mystery. It is the predictable outcome of ordinary biological rules operating through the asymmetrical circumstances of contact. Directional mating determined what entered the hybrid population. Selective pressures determined what survived within it. Social and demographic currents determined which traces endured. Together, these forces wove the same signature into every part of the genetic record. 1 to 3% of Neanderthal DNA scattered across our autosomes, none in our mitochondria, none in our Y chromosomes, and a marked depletion on the X where hybrid males once faltered. This is not a story of conquest or extinction so much as one of constraint. When two closely related species meet, evolution does not negotiate. It filters. Male-biased mating brought Neanderthal genes into the modern population through women who carried modern mitochondria. Male hybrid incompatibilities removed the Neanderthal Y. The remaining fragments of autosomal DNA persisted because they neither hindered nor helped enough to be erased. Each process reinforced the others. None stands alone. What looks at first like a puzzle of missing pieces is in fact a single coherent pattern of selective survival. And that pattern tells us something larger. The forces that shape the meeting of Neanderthals and modern humans, sex bias, social structure, and biological compatibility, are not relics of prehistory. They are recurring engines of genetic change. Across time, the Y chromosome has proven volatile, the mitochondrial genome stable yet replaceable, and the autosomes resilient recorders of distant contact. These asymmetries have replayed in every era of human expansion, where social hierarchies, mobility, and marriage patterns have guided the flow of genes as surely as geography or chance. In the end, what our genomes preserve is not merely the fact of interbreeding, but the structure of it. The the subtle choreography of who mated with whom, which unions produced fertile offspring, and which lineages multiplied afterward. The absence of Neanderthal mitochondria and Neanderthal Y chromosomes is not an absence of evidence. It is the evidence, a fossil of asymmetry written in the logic of inheritance. Through it, we glimpse the most intimate scale of human evolution, not battle or migration, but reproduction itself. It is there, in the quiet filter of viable unions, that two species met, mingled, and became one.